Welcome everyone. It's really my great pleasure to introduce Dr. Dennis Stevenson. I was hoping I could stand up here and say some really embarrassing story about him, but when I thought about it, I could only think of good things to say. That's the best I could do. So Dennis got his degree, his undergraduate degree at the other OSU, that little one in Ohio. And then he went on to get his PhD at UC Davis in botany, or was it in plant sciences? In botany. After a number of positions at spectacular botanical gardens throughout the world, Dennis settled down in New York City, where he was an assistant professor of biology at Barnard, at Columbia University, Barnard College. And then he joined the staff of the New York Botanical Garden in 1987, where he's been ever since. He has worked at a number of different positions there. He's currently the vice president for botanical research. I believe is that your correct position? That's what your CV says, you know. They change the name every now and then, so I get and at a position which you can see he is very enthusiastic about. He's also an adjunct professor at 6 different universities, which really speaks to Dennis's depth and breadth of knowledge and the desire for various students to learn some of the knowledge, to share some of the knowledge that Dennis has to impart. He's won a large number of awards and research grants over the years, and currently he's one of the PIs, among other things, he's a PI on the Plantiome project with Pankaj. I met Dennis when I was a postdoc. He hired me to be the postdoc, a postdoc on the Plant Ontology project, which is how Lowell and I got to know him and Dennis. And so I guess, I remember going into that interview just thinking, please don't let me say ontogeny. Please don't let me say ontogeny. And I didn't say ontogeny. And so I convinced him that I could do ontologies, and he hired me to do the job. And so that's why I'm here today. But more importantly, Dennis has introduced many, many scientists to ontologies. He is a marvelous ambassador from our community to the plant science and broader biology community. And he's also one of the best resources, I'm sure many of you know, that an ontologist can have, which is someone who understands their domain and the science behind it very deeply, and understands it in a way that he can communicate it to the ontologist so that we can build good ontologies. And with that, I'm going to let him share some of his experience about ontologies with you. Do you want to say anything about afterwards? I think that's okay, so we'll have some time for question and answers afterwards.
Great. Thank you. And as everybody, Dennis is transitioning, please feel free to tweet ICBO2016 hashtag. So, thank you.
Well, I think I need to quit while I'm ahead. But one of the really great things being at this OSU versus the other OSU, there's 2 other OSUs, by the way. There's one in Oklahoma too, is that this one still has a botany department. And that's, I'd say, great for the botanists. So I had a little sort of conversation with Barry this morning, which sort of made me think about this on the front end. What we do in biology in particular, I think, is we think in pictures usually. So part of our problem is making our language match the images that bring to mind structures to people across the plant kingdom. So I kind of want to use that as where are we, where do we want to go, and what have we done, sort of in a way. So one of the ideas for the plant ontology was to go, as the title says, to try to relate the Go terms, the gene ontology, to the plant ontology. And the overall notion here is that people could go get an image in the plant ontology, and that would track them right through to the genes that might be responsible for that structure that you were looking at, or morphological form. And the reverse would be true. Somebody working in the gene ontology that's working on a gene could look at that, and it would take them all the way through the structure they might see. And they would see that not only as a structure, but we would actually put the variability that's known to exist in that structure in the images. And we would also, as you'll see, I have a couple of them in here, we would also use line art, because line art is often really the best way to see something as opposed to color images. Nothing wrong with color images, but often line art causes you to focus on the particular item that you're looking at. So what we really want to do, as is shown here, is we want to take this thing on the left and relate it to gene sequences on the right. And what we want to do then, of course, is identify how those genes function within networks. So let's just set some stage. So I stole this, I think, from our website or whatever. Well, what I wanted to use this for was these diagrams really get to you sometimes. But but if you look over here, oh, I'm pointing at my screen. That's really good, isn't it? That's this. I don't want to get anybody's eyeballs. Okay. So I really want to focus on these items right here, what I would call characters as a structural botanist and a person who works in phylogenetics. And then, of course, we want to talk, those things are important as what we would call the phenotypes. And of course, the phenotype is not necessarily, the phenotype is something you see and you can put a descriptor name on. But what we're also after is the origin of that phenotype. And that origin can be very different. So you can get very similar things that have a very different history and therefore different genes possibly underlying building or contributing to that structure. Now, I just want to mention this because one of the things that's really pushing it forward for us on identifying gene to trait, in a sense, is this project, excuse me, called the 1KP project, where they did 1,000 land plant transcriptomes, and we went from the algae through the flowering plants. The focus was, a lot of the focus was on what we would call the mosses and ferns and these lower groups, because we know a lot about the flowering plants. But this gives you just an idea of how much data was produced in about 3 years. The other thing about this was, by having people who are more organismic biologists choose what we would call the terminals or the species that would be used in this, meant that the sampling was very good across all kinds of groups of organisms in terms of having the number of species as a representative proportion. So the 1KP has been very, very useful, and we're going to see at the very end some things that came out of 1KP that have allowed us to identify the genes, and now we'll be working with imaging the structures that relate to that. So we need to consider some concepts, and so I need to sort of go through these and in a way show you about these. So as I've already mentioned, there's a phenome. I'd love to use the term phenology, but it really means in a growing season when the leaves come out and then the flowers and stuff on a regular basis. So I use phenomes, but we know what genomes are and we know what phenomes are. And interestingly enough, the phenomes are things that we can observe, but genomes we can't. We can only observe them in terms of how they're expressed in making phenomes. But phenology, not phenology, but phenomics, I guess is what you call it. Anyway, that's the important part in an everyday life that has the broadest number of users. So how can we bring these different users with different ideas and different things that they want out of it together? And part of this issue is about evolution and change. So we're really looking at the genes that underlie the changes that have occurred in history. And evolution is about problem solving. So organisms have gone about solving their problems often with what look to be the very exact same phenome, but in fact actually have very different genes that give rise to that. And maybe the simplest one I can give you is, and I didn't put it, I should have used an illustration for this, but cacti. Cacti occur only in the New World. So the good old cactus you see is something you're going to find in the New World. The Old World has a counterpart in a group called the euphorbs, and they look just like cactus, but the parts that make them look like cactus are actually different morphological parts. So in a description, you're going to use the same kinds of terminology as for both because they look the same, but in fact, they're very, very different. And you wouldn't know that. If you were a plant systemicist, you might know it because they occur in very different plant orders. And we'll come to that because I will give some examples of it. The other thing that comes into play is something that I put in here called homology versus descriptors. Well, for phenology type or phenome type stuff, we want descriptors. That is a word that describes what we see in an accurate way. I'm going to use some examples of that. One of them is a term called ligule, which comes from the Latin. It means tongue-shaped things. And so that's, we know we can have an image of that. But homology is a different thing that biologists work at, because homology is something that biologists have, that when we make a phylogenetic tree that is a family tree of life, we actually take features on there and say, oh, this identifies this group of organisms, and so this feature is homologous and should have all the same genes operating to produce the same thing, as opposed to the thing that looked like that occurred in a different group. Now, it's very useful if you're a molecular biologist, a plant molecular biologist that does Evo-Devo, the evolution of development. I think Evo-Devo would be a great rock group name. But if you're doing Evo-Devo, you really want to have things that are homologous when you're looking at the controls and the things in the genome. You may be interested in a certain feature, and you need to know there's another taxon or entity that has the exact same feature that may be easier for you to work with experimentally, or may be more useful in an agronomic sense. So homology is an important issue here in the plant ontology, but being a descriptor is also an important issue. So where shall the twain meet is part of what this is about. And that goes to the next thing, homoplazy versus ignorance. So often because we use these descriptors, and then when we do these phylogenetic or family trees, we go, oh, look, it's not, they're in different places, but we call the same thing. This is terrible. It's homoplastic. It's uninformative to me. Well, that's because we are ignorant of what it really is. If we knew the genes and various things that underlie it and the fact that it occurs independently in different groups, we would then not call it the same thing. So we would get around this homoplazy. And that's just a thing that I love to do a diatribe on against molecular systematists who seem to think observations on living organisms are superfluous. And then the final kind of thing we want to come to is, how are we going to incorporate this data from genomes and phenomes into descriptors that people can use, whether they're describing plants in a flora or whether they're describing genes and the structures that go with them. So let's go down that path a little bit. So here's the descriptor thing. This is what people do. I do this all the time. You need descriptors to have keys to identify species, or in this case, families. And in a moment, I'll give you an example where I highlighted the things that you would have in an ontology. But you can see that there's different kinds of terminology in here. And so you can have keys to this higher order level, like your family. That's really your second name, but in that sense. Or you can look at individual genera or species as in the lower ones. So all we're saying is that we're going to have to use this word for descriptors here, and that's just where people pick the terminology up to try to understand if they're dealing with the same structure. Here's a description, and I just highlighted a lot of terms in there. These are all terms that we could put into the PO. The problem will be that some people don't use the same term for the same thing, or they use the same term for different things. I'll give you one example that you can find in here. I'm not going to go through these, but I use a term in here called strobili, which is, I can't see these, so I can't really point to them. It's down in here. There we go. Lots of people would use the term cone. So you need to, we have a case where we have synonyms, and in this case, the synonyms might be equal, but they're really not, because cones are often used for compound structures, and this group of organisms are used for simple structures. So then you've got to put adjectives on them. So we'll go look at examples of having to use adjectives to make the language straight and to make it more useful for everybody, but there's only so many adjectives you can put on the term. Well, here's one of the examples that I really want to talk about. It's a structure here in the bottom right called a ligule. And I already mentioned it. Well, if you look at this, this is just a phylogenetic tree. It's a bit of an old one of the land plants. Those circles on there, interesting enough, are places where we have not been able to resolve the relationships. In other words, if relationships, I'm going to have to use this one. If relationships are completely resolved, then you would have this completely dichotomous portion. If you have a place like this where there's several branches coming off without any kind of dichotomies, like here, then that means you haven't really been able yet to sort it out and terminate who's most closely related to whom. And those are the two nodes that are the big problem in phylogenetics. But what I really I think most of you see that way, but I've got to see this way. It's because I'm right-handed, probably. But look at this. This term ligule occurs 5 times in the flowering plants and 1 time down here in this little spike moss thing that out in the west here is mostly in the deserts. And we know that this group is distantly related to this group. And I'll just tell you, the 5 occurrences here are in groups that are not related to each other. So the occurrences in here, there's an independent use of the term ligule in the grasses that we see, 1 in the ginger relatives, 1 in the palms, and these are called monocots, which would be one of these kind of units if I'd put it in there. And then 2 times in these dicots, and these are totally unrelated organisms, which tells you that these things are developed independently there. So we'll just take a look at that. And I actually put the PO term in there, which we're going to work on. One of the ideas about putting things out like this and starting the PO and having terms is you can begin then to test them as hypotheses, and you can come back, of course, and flip-flop. I call it, of course, just recognizing data, but the politicians call it flip-flopping. I flip-flop a lot, especially if someone gives me, you know, information that's relevant. So let's take a look at a ligule. So the ligule is this little structure in Selaginella. They have little tiny leaves that are about like this. And on this surface called the adaxial surface, there's this funny little tongue thing that develops that nobody has any idea what it's for or why it's there. But you know what's really great about it is fossils have it. So it allows you to place fossils when you get them. This is just a low mag of it. Gives you an idea of the size of it. Compared to this thing, which would be kind of like a pollen sac in a flowering plant. It's also used in a group called the composites. This is a sunflower family, and some of the composites have this, but not all of them. Sunflowers would. This is an individual flower. This isn't a flower. It's an inflorescence composed of many, many flowers. So, you know, think about taking a lily plant with all those individual flowers and cramming them down into this condensed structure here. And it only has 1 petal left, and that petal is called a ligule. Well, I'll tell you, this petal is not homologous with this little funny thing on the sides of the leaves of some little lycophyte down there. Similarly, we have them in gingers here, which here's the leaf inserted as we saw in the definition, and there's this little process that comes up that's sort of tongue-shaped. So that's a ligule in that group. Here's the grasses, which have it in the same place in a way where the leaf blade expands from the lower structure, and here's the sheathing structure or kind of tubular growth that comes up that's also called a ligule. Now, the interesting thing about this is the ligule in the grasses actually defines that group. It's a homology throughout the whole group, and it can define it. The palm people, usually the palm people are a little out to lunch, but they got it together, and they actually call the same structure in a certain group of palms, the fan palms that used to be called a ligule, now called a hastula. So part of what we're thinking about here is, do we need different names for these structures in these different groups, or do we need to put an adjective on them? And the reason I say that, if you go into like your JSTOR searches, or we have something now called Biodiversity Heritage Library, greatest thing that's happened to me in my life in terms of trying to find old, generally irrelevant except to specialist literature. But if you do a search, if you're working in molecular biology, you go, oh, you know what, I'm interested in a ligule. I'll search ligule and find out where those guys are. You're going to get this mess of stuff that has nothing to do with each other. So if you're interested in sorting out the genes to give rise to that kind of shape structure, that's useful. But otherwise, you're going to be led down the wrong path and looking for things. So I want to take this in a real-time example. And that's partially because this is a group that is very dear to my heart. So we have a group here called the cycads, which is this unit in the seed plants. And we have a structure called a peltate megasporophyll. So it says megasporophyll shape. And you make a data matrix and you code the different terminals you have, which are all the guys here. And you code them for whether they have it or whether they have a flat one. I didn't put the picture of the flat ones in, but this is And if you look at this phylogenetically, and I know there's an alternate interpretation, but you'll see why I didn't use the alternate one. You can see that this structure occurs 3 times independently in this group. That's telling me something about this, that these are maybe not the same thing through time. That is, in history, they converged, right? Because it's the best answer to a problem they had of sealing their cones up. You needed to develop a structure that when you look at it, looks peltate. That is, this structure here, which we're looking at from the bottom out, looks like an umbrella is the way it is, with a stalk and 2 little seeds on it that are still there. So we have examples of this in those 3 units that appear to be now independent. Well, that's a testable hypothesis. And so if we look along here, and I'm just going to tell you this, and you have to trust me. And you have to trust me to push the right button too. Okay, so if you look at that in this group called the Zamia clade here, this structure starts out as a primordium that's a post and develops a ring of growth that gives it the umbrella shape. This group, in contrast, starts out with a post. This is the one down here. And instead of getting a ring that grows around it, what happens is it grows only on one side. It makes a big mound up and around a little bit. And because there's physical compression, the bottom bulges out so that you end up with a peltate structure. So we now know we have 2 different ways of achieving the same structure. We now know that we can look at the genes involved in developing this adaxial growth versus the ring growth, etc. And we therefore understand more about the biology of the organisms, and we have more effectively communicated with a broader audience. The same thing's true in what people call the molecular characters. So if you make a phylogenetic tree based on molecular characters, because they're supposed to be, oh, so much better, because these morphological ones have homoplasy and therefore could mislead you in the terminology, you don't have to look very far in here to start seeing things like this, positions in a gene sequence that are independently derived in the same place in different clades. So the same issue occurs in this molecular data, but you know what? You can't go back and test that hypothesis, because an A is an A is an A is an A, and it will always be the same. But what you know about it that's different through is that this is about time. So these are not the same A's. These A's were like T's or whatever they were. I didn't put them on here, and they mutated into an A, for example, in, or whatever I got there, C's, in this unit as well as in this unit, right? Because it's time. So those things, although they seem identical, in fact, have a different history. And it's this history that we want to utilize. So let's go back now for a moment, and I'm going to take a particular plant structure and deal with that, a structure called a leaf. And so this is your basic plant, and geez, I'm in a botany department, I don't need to do this. But here's our basic plant with a stem and some roots, and these structures called leaves here. And we all know if you whack the top out of a plant, it grows new branches out. And those always occur in the axil of one of these leaves and a stem as a little bud. Now, leaves can be simple leaves like this, but often leaves are actually what we call compounds. So we're going to just know, and they often have little structures at the base. You see, they aren't drawn in this one, but they are here, called a stipule. So these stipules are parts of leaves that may be there or may not be there, depending on the plant you're looking at and whether it develops those. And it usually has a little stalk on the leaf called a petiole. So we're just going to utilize those relationships to look at structures that we might have in the PO and how we might have to modify things as we move through time. But I also wanted to use the leaf because I want to talk about the gene ontology at the end and how those relate. So there are these structures in plants. And this is true. The reasons why you want to be careful when you handle a plant is because they have things called spines, things called prickles, and things called thorns. And they're not the same thing, but this terminology is used interchangeably, particularly in the lay world, but even in the botanical world. Plant taxonomists do not pay attention to the structures they have in front of them, which they could do, and they willy-nilly mix these things all up. So I want to show you what they are, because they're very different, and there's even some things that can probably be added to the PO here. So, because we've been working on those. So, in some instances, like this Fuki area, this is going to be the spine, and it's really the petiole. This is a young stage, so this little leaf's going to develop the blade part and fall off and leave this petiole behind. It's actually only the bottom half of the petiole. That's one way to get a spine out of a leaf. Another way to get a spine out of a leaf is like in a cacti here, for example, where you turn all of the leaves on those buds that grow out into spines. The leaf is vestigial. So there would have been a vestigial leaf right here, and the little bud grew out, and all of its leaves are spines. So here's two different origins of the term spine, which is why we segregated those in the PO. One is a petiole, and the other is a whole leaf. Another way, remember I said there's those leaves that are compound? In some plants, you get the spine structures as modified leaflets in a regular basis. So it'd be interesting if you're looking at an Evo-Devo way to understand that this thing's actually different than this thing is actually different than this thing. And then there are some plants like in these acacias or in this thing where those two little stipules I talked at the bottom are actually the spine structures. So each of those become the spine. So here's a third way to get a spine in a group of organisms. And in this one, it's really cool. I just wanted to show you this. This is an acacia with a very complicated leaf, and the two basal stipules get big like this and are really nasty, and they have holes in them. And there's the world's nastiest ants live in those. So they're sort of a plant protector. It's kind of like, I learned the hard way, they're kind of like fire ants. Now let's move to prickles. So these are prickles are on these roses, and prickles are not related to a leaf position. So the area between two leaves, like we might have here, for example, just gets all these prickles on them like a rose. And prickles may not be the same thing across plants. Here they are on internodes. Here they are on the back of a developing leaf of a fern. Here they are on the trunk of a group of plants that occur in Africa that probably do a very different thing. I don't know why they're on the roses. I think just so you can't prune them. This plant, they're there partially because they radiate heat away from the plant physiologically, because these guys sit in the sun. Same thing with the cactus spines. This guy just has them on one side between the leaflets, and then these guys have prickles in between and sometimes both on the leaf braces and the stems. My point is this is occurring in very different groups of organisms, and we therefore might expect to find gene ontologies, for example, that are quite different. Thorns. Thorns are whole shoots. So they're whole little axillary buds that get nasty, like we might see here. Here's a leaf, and here's the structure in the axil. In bougainvillea, there's the leaf with the structure in the axil. This is a modified inflorescence, or group of flowers that just sort of starts down the road and stops and becomes this structure. So it's kind of interesting to know what might control an inflorescence aborting as it goes along. Because if you're thinking about this agronomically, you may want to cut down on the numbers of flowers and fruits on a plant to up all that photosynthetic production going into fewer units as opposed to many less nutritive units. And so these, oh man, that was, so could we do, yeah. So here they are in these things are ornamental street trees. They're called honey locusts. Most of them have had this bred out, but these are these branch systems. And you can even see little, you can't see them here, but there's little scale leaves if you look with a hand lens on here. So we have a whole different way now using the shoot system to do this. And how does that relate to modifying leaves to modifying the whole shoot system? And then the last one is this one, which we haven't dealt with very well either. And that is these things that are called spines on the palms. These things, But if you look at this one, there's a root cap on it if you're a plant anatomist. These are actually the roots that often, you know, when you cut a plant off and you stick it in some dirt or water and it forms a bunch of roots, they're called adventitious roots. Well, they have these adventitious roots that are actually turned into spines, particularly in the palms. And this is just the young ones coming out at the leaf nodes. So these are very useful often for some of these plants are climbing plants, and they sort of try to climb up, and this just hooks them on to the plants they're growing on. But regardless, we have roots doing the same thing. So you have to ask yourself, are the same genes there for sclerifying organs, or are there a different set of those, and can we utilize that information in some way? I want to talk about tendrils, because that's the other thing that's really, really interesting and actually of economic importance. So tendrils, as shown here, can either be this. You see, this is a leaf, and then an axillary bud, and then it turns into this tendril thing, right? That sort of coils like that, and it has a sort of behavior. We won't talk about that. Sometimes it's the end of a shoot that becomes a tendril. In this case, it's a bottom leaflet that becomes a tendril. So the main ways that you get those is whole leaves become tendrils, axillary buds become tendrils. There's a parallelism here, isn't there? Or parts of leaves can become tendrils. And these are, you know, effective in climbing. So you really want to know how tendrils are controlled, for example, in an ornamental plant that's of economic value versus our good old buddy, the grapevine. And if you look at that, we've sort of begun to deal with this by using adjectives on the fronts of them. So these are the things I described. And in the PO, we've now done all this. So now, given what was done in 1KP, we've begun to be down the road to the position now to identify the genes that would relate to those structures. I want to talk about now the notion of use of the gene ontology relating to the plant ontology. This is sort of a diagrammatic thing about what we know about the molecular genetics of leaf development, as it's, I know you're literate, in angiosperms. And there's a lot of genes and gene networks involved here and whatnot, but there's a couple that I want to focus on here. And one of them is what is called the NOX genes. The other genes that I want to briefly mention are the HD ZIP3 genes. These are what were called gene families, and the CUP genes. And these are involved in the identity of whether you're going to be a shoot and maintain your identity as a shoot by a growing apex, or whether you're going to become as a lateral product of that, because you could be a branch if you worked with the NOX, or whether that lateral product is going to end up being a leaf-like structure or flat structure. So we actually have some data on these now, mainly due to 1KP. So let's take a look at that a little briefly. So there's a recent paper out on this. So generally speaking, our good buddies, the ferns, so this is what's called the shoot apical meristem, sometimes called the shoot apex. Those terms are straightened out in the PO, but sometimes old people like myself haven't adapted to it as well as they should have. But there's a cell up here that was called the apical cell. And that was considered to be the shoot apex of the ferns versus a whole region like this in the higher plants, that is in the flowering plants and the conifers and things like that. It ends up that if you begin to look at this with in situs and gene expression and eventually doing some knockouts, that's not the case at all. There's that flowering. I want to make, I guess, a second point. I'm getting a little, I don't want to have epiphanies when I'm talking, but basically, in the world of Evo-Devo, all of our thinking is from 2 or 3 model, and by the way, they're not model organisms, they're species. Organisms are individuals, but of the model species down. So the original work was on Arabidopsis because the geneticists loved it, easy to work with and grow, low chromosome number, those sorts of things. And so we had a few flowering plants, and then we tried to extrapolate that down the tree of life through history, when in fact we had no data down there at the bottom. We didn't have any information on the ferns. We didn't have any information on the mosses and very little on the gymnosperms. So that really wasn't working very well. Yet we had classical interpretations that told us, well, these things are different. But actually, that set up a testable hypothesis, didn't it, to look at those differences. And it's really interesting because now there is a lot of focus on those. For example, hornworts are having their whole genome sequenced. Some of the bryophytes, I just got some money to help the people do fern genomes. You know why people don't do those plants, some of them? The genomes in the gymnosperms and ferns are massive. You know, Arabidopsis is like 6 gigabases or something like that. Gymnosperms are like 53. So part of it is a finance problem, and part of it is the problem of trying to analyze and put together those genomes. But anyway, with that aside, let's just go back to what I was talking about here a little bit. So if you do in situs, it ends up that the Knox genes are spread throughout this area, but not in this thing that was called the apical cell. So if you take this diagram here, it's a good example. This is where most of the Knox genes are being expressed, giving rise to the cell division that allows you to maintain a dome-shaped structure through time. So we know now that we can relate the Knox genes in the GO to the shoot apical organization in the PO. And these are just diagrams because these are based on ferns of these gene expressions. The other thing that's really peculiar with these in situ things is they're often expressed in the vasculature for the people that know that kind of structure. Okay. Now, I just put this in. This is about the class 3 zip genes. So I'm actually going to zip back 2 of them. And what we want to talk about is the class 3 zip genes and more or less this group called CUP. And the zip genes give you the identity, what we call, they're expressed in the adaxial or this part of the leaf to cause this sort of flat structure to thicken out a little bit. And then the presence of the zip genes and the CUP genes actually allow you to develop the marginal growth for the flat leaf that you see. So it goes from a post into this flat structure. So you really need both of those working together. And if you do a knockout, what people call the knockouts on these, in other words, if you knock out the zips or you knock out the cups, the leaf never develops. It just sits around as a little skinny post, and that's that. So that's really not going to help you in the photosynthesis world. So let's just take that and look at this. And it's really quite interesting. And I don't, I'm not going to do a bunch of detail here, but if you look at this, one of the things that's gone on in the history of the land plants, you see here we got the mosses and the lycophytes and our good buddies, the ferns and the seed plants and the flowering plants, which are frosting on the evolutionary cake. Anyway, if you look up through here, what you get are duplications of these genes. So once you get a duplication and multiple copies of a gene, you can do strange things with that, which really is going to intersect with the PO. And what do I mean by that? It means that when you have there are 2 things about this. When you get a genome duplication, then you duplicate all the genes in the organism. You make extra copies. Usually a diploid should have 2 copies to start with. Those are available to be co-opted to make other structures. So there's a problem we have, I think, in relating the GO terms to the PO terms sometimes in terms of what we call orthology. An orthologous gene is the one that's basically I guess the actual one's probably around from ancient times, but my point is the orthologous gene is the original copy that's doing the original things, and the paralogs are the ones that were duplicated that are available either as backups in case something happens to the actual functional one or to become other things. And we know that they become other things. So the class 3 zip genes, though, the really important thing is they go all the way back in land plants to saying that all the leaves in the land plants are using class 3 zip genes plus the CUP genes. So the leaves in the land plants are homologous structures at some level. And we don't have to think anymore that the lycophytes had a different origin because the paleobotanists kind of do that with the fossils, because at least in the extant plants, we know that that's the case. So that's a valuable thing, I think, in terms of linking the geos to the POs and straightening things out. Here's another one that I think is really, really interesting that we stumbled onto, some people did a while back. And there's a group so this is mostly flowering plants. Some of the other guys are down here as the out groups. But there's a group of flowering plants called the monocots, which economically are probably one of the most important groups of plants we have, because it's all the cereal grains, all the rangeland grasses, the palms, great things like the yams that some people really like, and lilies and stuff like that. Well, they're called monocots because most plants we think of, they're called dicots, means cot is for cotyledon, and di means they have two seed leaves. You know, when you see a bean plant germinate, or when you take a peanut apart, when you take it apart, you see the little shoot inside. Well, those two things that you eat are the cotyledons. So they're dicots. Monocots have one cotyledon, supposedly. That's why they're called. They don't have any cotyledons. And we know that a little bit because out of the 1KP data and other data, the monocot clade has these cell commitment genes that whack out the development of cotyledons. And the result of that is that the monocot thing that's supposed to be the cot is the first leaf and uses the same cup genes and the HD zip3 genes and has these things that just knock out the cells that would be the normal cotyledons in that development. So I'm getting at this somewhere. So now we know we have something that says there's a homology here. In this case, it's like the lack of something. In other words, in the PO, do we want to develop some terminology about things that are not there, that are lost, genes that cause things to be lost? Because when we think in these phylogenetic trees, for example, when we think about this, the other things that help support some of these groups, okay, you might have flower that supports a group, but inside of there, you might have a group that lost their roots that supports a group. So if you know there's a lost thing that you can identify, it means the people working in Evo-Devo have a comparison between the things that have and between the haves and have-nots in a real way. So we hook those up, and we've done what we want to do. So maybe there's some things to talk about at the end. I know I'm going to get done early, but one of the reasons I wanted to do that was maybe we can have some discussions about how to handle some things. So we've got a long ways to go on this. That should be pretty obvious. And we have some things here to do. And one of those is dealing with what we call synonymies. So the synonymies, there are two kinds of synonymies in a way, but I think Barry would say there's only one kind of synonymy. In taxonomy, a synonymy is when you have two different names for the same species. So I think in morphology and the plant ontology, synonymies would be when you have two different names for the same thing. But the other synonymy that exists out there is having the same name for very different things. But yet they're always considered the same. So that in a sense, it's taxonomic. It's a different kind of way of viewing that. But we have to deal with that. So we have to work those out. And we have to try to work those out by combining, for example, some knowledge of the gene ontology with the plant ontology. The current and future directions of where we're at, in a sense, is that we need to begin to do things like more and more imaging. So my goal in the next couple of years is to do nothing but image plant structures in the way I talked about in the introduction. In other words, I need to image all of these sort of synonymy type things, number one. And number two, I need to show some variability in that. It's like, you know, we're all of one species here, but we can identify each other as entities. So in a way, you have to be able to incorporate that kind of variability in understanding your plant structures. The other thing that we need to do, which we're doing more of, is mining the 1KP because it's grown. We've now added 400 more species to it, etc. So we need to mine that in terms of trying to find more of the Go terms. And conversely, we need to mine the Go terms in terms of what people think they know and apply those to the plant ontology. So we're at a point now, I think, where we can do informed decision making on how we're going to move forward with this. And what's the end product here. The end products are developing this vocabulary of plants that can be useful from the molecular biologist to the plant taxonomist. Problem is the molecular biologists don't have the vocabulary, so they don't care what you put on there because they're going to be happy to have something on there. It's going to be the plant taxonomists and anatomists that are going to be the recalcitrant group because, well, we always called it that, but you know what? I grew up on a farm without indoor plumbing. I don't go, oh, well, I always went outside, especially in the winter. So, I mean, we've got to get past some of those kinds of thinking and begin to combine things so that everybody communicates the same things to everybody else and using their sort of visual images for it. So with that, I'm done. So thanks. Wow, that disappeared.
Thank you very much, Dennis, for your wonderful talk and sharing your insights. We have time for some discussion and questions, so I welcome people to come up to the microphone, and there's one on that side and there's one on this side.
So I'll have a go with lacking roots. The relation ontology has a lax relation, and we could relatively easily add the lax relation to the plant ontology alongside part of and has part and derived from and is a and so on. But the question is to decide whether it's needed. So the reason why we needed the lax relation is because we sometimes need to say things like, because of this mutation, this patient lacks a nipple or lacks a finger.
Mm-hmm.
So in other words, we use the lax relation when there should have been something there, but it's missing. So we don't say, you lack a concrete boot, or you lack a steel girder heart container, because you shouldn't have had any of those things in the first place. But we might say, you lack hair a bit. So in the plant world, do we want to say that these plants lack roots because they should have had roots? That doesn't sound quite right to me.
I would answer that, Barry. There's two ways for that. There are things that lack them because they shouldn't have had them because their ancestors never had them. But then if you look up inside, if you had so that would be true for like the moss. But if you looked up, at the crown group there, the flowering plants, they should all have roots. And if they don't have roots and can't develop them, then in a sense they're lacking roots. And that means something has said, you're not going to develop your root. So from the developmental point of view, that would be interesting to be able to record in some way.
So I guess there is another dimension here. So there are two parts to the plant ontology. There is the plant anatomy,
right,
and there is the plant development stage.
Right.
Now, the plant development stage is an ontology to describe the development of a single plant. Maybe we need a third ontology which would describe the development of plant species through evolutionary time. And then your lax relation might play the sort of role that you want. But it still doesn't sound right to me to talk about an individual plant lacking a root because one of its ancestors had a root. Well,
I think, I'll come back to that if some.
Sure. So a lot of the terms you've described have been based on what things look like. And that makes sense because that's what was available to most people trying to come up with terms. But you've described what's been learned a lot about the processes that generate those structures. So in the sorts of ontologies that I've worked on, we found it very useful to start with the process itself and then describe the outputs to those processes. So my question is, as you think about how to name these things and distinguish them, have you thought about defining them by the processes that generate them and sort of incorporate that into your terminology, your ontology?
I think, the way I think about that, that's an excellent point. So I'm in a position where I've got sort of two different things I need to do. I need to have these descriptors that people can use in floras and keys, because those are the most useful products I ever have. People are going to key psych eds out in Colombia using those keys for years and years, and that's how I find new species. But on the other hand, I want to have some accuracy to what you say about the development. So it may be in the end that we have to deal with the, as Barry brought up, that the plant anatomy thing is a descriptor vocabulary, and the plant development thing is the one that tells you how those descriptors came to be. And you might actually have something where the descriptor terms might actually have different pathways in the development term, in the development one, that sort of converge to the descriptor. So if you went to the descriptor and say, okay, there's 2 ways to get here, or 3 ways to get here for that piece of vocabulary that you're using. Because the people identifying plants don't care. They just don't care, you know. They're like, I don't know, they do a lot of ecobabble and stuff, and they go out and key them out, and there's no reason they should care and learn all of that.
So the process that you're talking about is the evolutionary process. So in order to relate these different structures which have the same function, the process that has to be captured somewhere in the ontology is evolution.
Right.
And so in that sense, that is the process. The molecules that make it up don't capture that knowledge.
Right. If I might have one of these keys, I probably should have used a flowering plant one, but I might have the lead might say flowers red, flowers white. Bingo, I've got a red one and I deal with the red, but there are different ways to get red flowers. There's the anthocyanin way and there's the betalain way, for example, which are very different chemical compounds in different groups.
And the same thing that shows up in all organisms. So crustacea have these legs, which have sensors on the end that are have evolved because of leg duplication, right?
Yeah.
So the same thing happens over again. But the reason you think that way is because you're thinking like an engineer. An engineer has many ways to build a carburetor out of many physical things, but all carburetors have the same function.
Yeah,
So,
that's a good point. Structure versus function.
And what we do when we think about evolution is we think about, one way of thinking about evolution is thinking about evolution as the blind watchmaker or the blind engineer. And so we think of things achieving functions, and those functions can be achieved in many different ways, the way engineers can achieve the same function in a device many different ways. And that's what needs to be captured here, I think, is that way of thinking.
Yeah, right. I agree. I just don't know how to do it. That's why you're here, to help us learn how to do that. Okay.
Thank you very much, Dennis. It was wonderful.